So
Okay, very well. So okay, tell me topics that you find difficult in biotechnology. First, we'll go through those, and then, as we've decided, we'll go through some previous year's important questions. What do you find difficult in this chapter? Atif, Fiba, this is so this cannot go without your discussions, right? Please feel free to write in the chat. Okay, what topics do you find difficult in biotechnology?
I think really this chapter is not very difficult personally.
Okay, so let's Hiba, what about you? So I will go through the chapter once. Hiba, you can write in the chat if you feel any topic that is difficult, okay? Okay, so what all arenas? So I think to Atif it is clear. So Atif, would you like to talk about the two core techniques on which the biotechnological principles are working? Like mostly what are these two core techniques that holds true for all biotechnological applications?
Yes, sir. So the two core techniques are genetic engineering and bioprocess engineering.
Mhm.
Genetic engineering involves techniques to modify the chemical composition of genetic material and
Mhm.
introduce into the host organism to study its phenotype. And bioprocess engineering is the maintenance of sterile maintenance of microbial contamination, that is sterile ambience in the chemical engineering process.
Okay, so one question which can be asked is it possible to call something a biotechnological process where without doing any genetic engineering, we're just doing bioprocess engineering? Or is it not possible?
It's not possible.
And think again, why is it not possible? Is it necessary that every time to do something using biotechnology, we have to tweak the genetics? So, what happens in case of IVF? Atif, because we listed it, right, as one of the biotechnological applications. What is IVF and what happens in it? Would you like to just summarize in one sentence?
Yes, sir. IVF stands for in vitro fertilization. This is like a type of fertilization in which a sperm is taken from the father and egg is taken from the mother, and they are both fertilized in a environment outside the body, in a fertile environment outside the body.
Correct. So sperm and egg are taken and fertilization is in vitro outside the body, right? Are we in this process? Are we tweaking? Are we doing any genetic engineering?
Yes.
All right. What are we doing? So,
Process engineering.
yeah, we are just creating sterile ambience, contamination free, where we can make these gametes fuse with each other and fertilization can happen. And when we are sure that the fertilization is successful and it becomes an embryo, starts dividing, then we can put it back into the female's body, right? Into the womb, basically. So yeah, it is possible to do bioprocess engineering without modifying the chemical composition of the genetic material. Okay. Make sense?
Yes, sir.
Good. Okay. So what is restriction enzyme? What do we understand by REs?
What is it?
REs, restriction enzymes. We're just going through the main things.
Yes, sir. So restriction enzymes is mainly it's a type of enzyme found in prokaryotes which can cut DNA.
Mm-hmm.
There are two types, that is restriction exonuclease and restriction endonuclease.
Mm-hmm.
Restriction exonuclease cuts DNA from the ends, while endonuclease cuts at specific sites between the DNA. And endonucleases are the ones that are used for recombinant DNA technology.
Why not exonuclease? Why can't we use exonuclease for a biotechnological application? Is it like we can and we are not using it because of some reasons, or we cannot use it? It is not useful at all. Mm-hmm. So this is not directly in the NCERT, but you have to think. That's a concept. Come on.
Sir, I might have an idea, but I have
Mhm.
like
Idea is good.
question about this.
Idea will work. So tell me your idea and tell me your question.
No, so the question is, can plasmids be cut using restriction in exonucleus, or can they be only be cut using endonucleus?
That's what I'm asking you. That is exactly my question indirectly. Because in biotechnology, we heavily depend on plasmids, right? So my question is, why do we only choose endonucleases, not exonucleases, to work with for any recombinant DNA technology? Like, do we choose it because we prefer one, but the other can also be used, or the other is just useless?
Yes, so because exonucleases cannot be used to cut plasmids, like plasmids are circular and they have no ends, so we can only use endonuclease to cut them.
Perfect. Yeah, right. Makes all the sense. And I think so. And also, exonucleases do not cut, right? They only like remove base by base from the ends. So they're like chewing from the ends, degrading the DNA from the ends, right? Make sense? Yeah. That's why only endonucleases are useful, because they cut at specific sites. Okay. And what are these specific sites? What are palindromic sequence here?
Yes, so the palindromic sequences are the like each restriction endonuclease has like a special palindromic sequences that only it can recognize, and it can only cut through this sequence. This is the only sequence that it can recognize, so it's known as the recognition sequence. A palindromic sequence means like the palindromic sequence means it reads the same from both ends. For example, in this one, if you read from 3 prime to 5 prime, it reads as CTTAAG. It can also be read on the other strand as in the same way. And then so endonucleases can either leave like blunt ends or sticky ends. Sticky ends means they like cut a little away from the center to produce like overhanging ends called sticky ends. Blunt ends means they simply cut it in a straight line to produce
So,
ends. Yes.
do you think so? Do endonucleases also leave blunt ends?
Yes.
Yeah. So, in what case? In what cases can they leave blunt ends?
Some endonucleases leave blunt ends, some leave sticky ends.
Yeah. And what? So, when will they leave sticky end? When will they leave blunt ends? Because the sequences can be designed. So it's always designed in a way for experiments where there's no blunt end left. So how do we ensure that they don't leave blunt end? Because if they leave blunt ends, they're of no use to us, right? Or I should say they're of less use to us.
Yes, sir.
Mm. So we ensure it by making sure that the recognition site, let's say if it is between two AA, in this case, between adenine and adenine, then this adenine and adenine should not be in the center, because then it will be a blunt straight cut, right? If they are slightly away from the center towards the right or the left, then they will not leave blunt ends. Then there will always be sticky ends because of unbounded or free nitrogenous bases hanging, right? Which can make hydrogen bonding. The whole idea is to, So if this is a slab, don't cut it like this. Yeah. Instead, cut it like this, right? So that this one piece and this one piece are two kind of overlapping sticky ends, which I have shown here, right? You understand?
Yes.
Yeah. Okay. So that's how we ensure. Okay, a quick review of agarose gel electrophoresis. Hiba, if you can speak, that will be great. If you can contribute to the discussion, are you there? No, I guess. Okay, Atif. And it has to be you and me.
Yes, sir.
I'm saying that it has to be you and me. Hiba, I think, can't speak up. So, a quick review of gel electrophoresis, agarose gel electrophoresis.
Sir, can you explain this one? This one I don't remember.
Yeah, okay, great. No problem. So, I remember I said that the first thing is we need to cut the DNA, right? But when we cut the DNA, the problem is that there are various different sizes of DNA fragments that will be present in our solution, right? Because we took the whole genome, the whole DNA of the organism from that cell, and we cut it using restriction endonuclease, right? So the problem is how to select our gene of interest, the fragment that is our gene of interest. For that, it is a separation and isolation technique where agarose is a biological polymer which is extracted from seaweeds. And gel electrophoresis is it solidified, semi-solidifies as a gel. And electrophoresis simply means to separate using electricity. Now, why it works in the case of DNA is because DNA is negatively charged, correct? So we keep this gel, which has these wells, like holes, in a buffer, which is a electrolyte buffer, and attach a battery to it, electrodes. So negative electrode and positive electrode. Okay. So DNA will move from negative electrode because it's negatively charged. We load it towards the negative terminal. So it will start moving towards the positive terminal. Make sense? And in the first well, we are loading a reference ladder, which will tell us that if the band comes at this position, it will be this big. This will be the length. So the smallest fragment will move the farthest away from the negative terminal because it's small and it can easily move up through the pores of this gel, right? We discussed this pore sizes, right? And then we can take that part, elute out the DNA of interest, and we get our gene of interest. Make sense?
Yes, sir.
Yeah. So that's gel electrophoresis. Agarose is the polymer. Okay, now this question is a direct question from exams. what are the features required to make a good cloning vector? Yes. So there were three we discussed. Atif, would you like to go over it?
Yes, sir. So there are three features that are required to make a good cloning vector. The first one is origin of replication.
Mm-hmm.
So the origin of replication in the DNA, it controls the copy number. So it will replicate more. So the origin that supports a high copy number is preferred, so the DNA can be amplified easily.
Mm-hmm.
And the second one is a selectable marker, which helps in identifying and eliminating non-transformants.
Mm-hmm.
Example of this is the genes for antibiotic resistance.
Correct. How does it help to identify?
So I'm not sure. I don't remember this one.
So think
So we
about
can
it.
take the
Right.
example
We
of
have. Mm-hmm. Mm-hmm.
I think ampicillin and tetracycline. We can take these two examples.
Okay.
Like we can deactivate these genes to insertion activation by uh, cutting cutting these genes for, like, for example, if the it's like so there are like recognition sequences in this ampicillin and tetracycline, and if we cut the gene at that part and insert the gene of interest, it will deactivate these genes. So
Right.
this is known as insertion activation. So for example, if we deactivate the gene for tetracycline, then if we grow it in a medium of tetracycline, then the the ones in which the gene has been inserted, the transformants will not grow and the non-transformants will grow. So like that, we can differentiate between transformants and non-transformants.
Right. So that is a negative way to go through it. But insertional inactivation is, I think it's mostly useful in case of beta galactosidase. But when you have antibiotic resistance, the idea is that why to then deactivate this gene? Just put a antibiotic resistant gene so that only the transformants can grow in that culture plate containing that antibiotic. So let's say if I have ampicillin resistant gene in my plasmid. So if the bacteria has taken up the plasmid, the bacteria also now has the antibiotic resistant gene inside it, which will be replicated, transcribed and translated, produce the protein that will give ampicillin resistance to the bacteria, right? Now, those bacteria that will not have the plasmid will also not have this antibiotic resistant gene, and they cannot survive in ampicillin environment. So instead of deactivating ampicillin, the straightforward way is we insert our gene of interest somewhere else, which does not deactivate ampicillin resistance, and then we check the bacteria on a agar plate containing ampicillin, right? If it grows, it is a transformant, which is our bacteria that we need. If it doesn't grow, anyways, it was not useful to us. So this way we will only get the bacteria growing that has our gene of interest or our plasmid. How about that? Does it make any sense?
Yes.
Yeah. Yeah, and the insertional inactivation works in, so this is another way through which we can select cells where the idea is not to use any antibiotic resistant gene, because the problem is the more exposure of antibiotic resistance to bacteria is also not good, right? So to not use antibiotics, what you can use is a colory enzymatic reaction which produces a color. So there beta galactosidase is a better selectable marker gene. So beta galactosidase, when is present, which means it is not inactivated by inserting our gene of interest in between, then what happens is that beta galactosidase will be produced. It will react with substrate in the environment and will produce a color. So if you get the color on the of the colony on the plate, you know it is not transformed, right? But when you insert the gene in between the beta galactosidase sequence, so beta galactosidase gets kind of inactivated. That's called insertional inactivation. What are you inserting? The gene of interest, right? And what are you inactivating? Beta galactosidase. So now this won't be able to catalyze the reaction that produces color. Now the colonies will be colorless, just a normal colony. So if you get normal colonies, you know that, you know, it does not have our gene of interest. Make sense?
Yes, sir.
Yeah. So if we are anyways using antibiotics, we don't need to do insertional inactivation unless we are using a combination of two antibiotic resistant gene in the same plasmid. Then we can inactivate one through inserting our gene of interest and can check it on a plate. Okay.
Yes, sir.
Yeah. And what is the third one? Cloning site.
Yes, sir. So the one is cloning sites. So a vector should preferably have like a single sequence, like two sequences for each recognition enzyme. Otherwise, it'll complicate the gene cloning.
Right.
Yeah, that's all I know.
Right. It won't just complicate. Um, how exactly will it complicate? What do you think?
Restriction enzyme, then you can cut out all those parts, which will result in a lot of fragments, which can be problematic.
Which is of no use, which is a useless. It's a linear fragment then. If anything is circular, this plasmid, and if I cut it at one place, it becomes linear, right? And then we insert our interest gene of interest and then ligate it back to make it circular again. But if there are two sites or three sites, basically we'll end up with 1, 2, and 3 fragments of no use, right? Yeah, so preferably just one. Okay, cool. PCR. What is it?
Yes, so the full form of PCR is polymerase chain reaction. It is a method of amplification usually, and it is used for usually diagnosing certain diseases or pathogens.
Mm-hmm.
So it contains of three steps. First, we take the beginning step is first we take a sample of the DNA to be with the region to be amplified. Then for the first step is we denature it. We heat it to a high temperature like 95 degrees Celsius, which separates both the strands.
Okay.
Next is annealing, in which we add RNA primers to the amplified region in order to replicate it, which is this is done as 72 degrees Celsius. Then next is we do a replication using a special type of DNA polymerase known as Taq polymerase. This is extracted from a bacteria known as Thermus aquaticus, and it is stable at high temperatures. So while
Mm-hmm.
it is kept at high temperatures, the denatured DNA is then replicated, and this process is known as extension. So with this process, we can amplify the DNA up to a million times if we repeatedly do a lot of cycles.
Yeah. Perfect. Okay. So rest is, I think, simpler. Let's look at some questions.
Yes.
Take 2-3 minutes and go through the notes. By the time I'm figuring out. Okay. Anything to discuss? Atif? Okay. So let's go through. So write your correct options in the chat just to me. Can you see the screen? Okay, so let's start with first one. Would you like to read, Atif?
Okay, sir. The DNA fragment separated on an agarose gel can be visualized after staining with option A acetocarmine, option B aniline blue, option C ethylium bromide, option D bromophenol blue.
Yep, in the chat. Correct. Atif. Hiba, we are waiting for you. Correct. Okay, now let's go to some difficult ones comparatively. Yes. 4. So I think I'll put a 2 minutes gap. It's not necessary for Atif to read all. Um, just read it and let me know. If you need a discussion, you can just write that you need a discussion. Correct, Hiba. Atif. Correct. It's A. Okay. 6. Correct. Atif is correct. Eva? Yes. Correct. Okay. 13. Yes, that's correct. Atif. Hmm. Okay. Why do you think that is the answer, Atif? Oh, what is your rationale behind it?
Yes. Sir, I knew that AB was like part of the process, so I thought it was D.
Sorry, your voice was lagging. Come again?
Yes, sir. I knew that A, B, and C were part of the process, so that's why I
Yeah.
like
Right.
choose.
So this is also a way to mm, like track down, right? How are ABC a part? Would you like to just quickly? So C is simply PCR, right? And that's very important whenever you do any DNA testing. How is A and B important or required? So the right is zinc finger analysis. Okay, Hiba. How is A and B required?
Yes, sir. A is required because we need to cut the DNA in order to check it. It's like very important. Yeah, we need to cut the
To,
DNA in order to actually check it properly.
to
I
sequence
think
it
the finger.
simply. You need to cut the DNA to sequence it, right? In the
Yes, sir.
fingerprinting, we are looking at small satellite DNA sequences. So we need it. We need the um, small, we need restriction enzymes to cut it into fragments where we can then sequence those small satellite repeats. Simple, right? And why is B important?
Yes, sir. B is required because we need to compare the sample DNA got with the one from the crime scene or whatever area of concern maybe.
Or from the the person, the donor who is being
Yes.
tested, right? Yeah. Okay. Mm, then the one that I chose was, yes, 19. This can you read, Atif?
Yes, sir. The colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria because of option A, insertional inactivation of alpha galactosidase in recombinant bacteria. Option B, inactivation of glycosidase enzyme in recombinant bacteria. Option C, non-recombinant bacteria containing beta galactosidase. Option D, insertion and activation of alpha galactosidase in non-recombinant bacteria.
Yeah, we just discussed it. Think about it. Yeah, that's correct, Atif. Yes, right, Hiba. Uh, why not B, Atif? Why you selected C over B?
Yes, sir. We know that the enzyme that produces the chromogenic substrate is beta galactosidase. Yeah, C is the only option that contains beta galactosidase. So I chose that one.
Hmm? So what's wrong with B?
The B is another enzyme, glycosidase.
Mm, yes, it's not glyco, it's galacto. Correct. And this is simple. Yep, 26. You wanna read? Okay, let's not read this. Just read it yourself and tell me. Yes, right. That is Atif. Correct, Hiba. Perfect. Now some convoluted questions are yes. This one. 38. Correct, both of you. So it was simple, I guess. Yeah. So 41. So it's very simple, just a little twisted. Will you read it, Atif, please?
Yes, sir. In genetic engineering, a DNA segment gene of interest is transferred to the host cell to a vector. Considering Consider the following four agents, 1 to 4 in this regard, and select the correct option about which one or more these can be used as a vector or vectors.
Mm-hmm.
Um, 1 bacterium, 2 plasmid, 3 plasmodium, 4 bacteriophage.
Yes, right, Atif. Um, why do you think, Hiba, bacterium can be a vector? Can it? Can bacteria be a vector, Atif?
So, did you ask something?
Can bacteria be a vector? What is the difference between a bacteria and a plasmid?
Yes, sir. A plasmid is basically a small circular segment of DNA in a bacteria's organism.
Yeah. Well, where are plasmids generally found?
In prokaryotes, like
Yeah,
bacteria.
exactly. Right. So the whole bacteria cannot be used as a vector. It's just the plasmid, right? So it has to be D. Does it does it make sense, Hiba? Okay. 49. Any confusion here, people? Is there any confusion regarding this? Atif, Heba. Yeah. So Heba wants an explanation. So basically, in recent years, DNA sequences are like basically nucleotide sequences, empty DNA, which is the mitochondrial DNA, and Y chromosomes were considered for the study of human evolution because. So, what is the what is significant about mitochondrial DNA, Adif? There has to be something about mitochondrial DNA and Y chromosome that is special. Atif? So, mitochondrial DNA is we only get it from mothers. It has a maternal inheritance lineage. Y chromosome we only get from like fathers. Only males get it from fathers. So they are uniparental, both mitochondrial DNA and Y chromosome in origin, uniparental, and do not take part in recombination. Okay. So that's why they are considered important for the study of human evolution. Make sense? Make sense, Atif? Am I audible? Hello. Hello, I'm audible. Yeah. Okay. Thank you. Heba. So is it clear why mitochondrial DNA and Y chromosomes are uniparental and they do not take part in recombination? Okay. And I'll take a few. This one. 57. Atif, are you there? Can you read?
Yes, sir. Sir, I have a doubt on the previous question.
Yes, let's go there first. What's your doubt?
Yes, sir. You said that Y chromosomes do not uh, undergo recombination, right?
Yeah, they're the smallest. Y chromosome is the smallest chromosome, which just contains around a few hundred genes, and it is always paired by X chromosome, which is very big. Now, remember, recombination was an event where uh, one chromatid, non-sister chromatid of one chromosome overlaps with the other non-sister chromatid of other chromosome.
Yes, sir.
But if the Y chromosome is tiny in front of X chromosome, there won't be a recombination possible, right? Because the physical crossing over is not possible between Y and X. And that is why males stay males and females stay females. Otherwise, if crossing over between X and Y could happen, some Y genes will just come in the X chromosome over time, right?
Yes, sir.
Yeah. So Y chromosome is very small as compared to the X chromosome. So homologous chromosomes concept does not fit on X and Y. Homologous chromosomes are those which have same size, same length of the arms, and same genes present at the same locus. Correct? Yeah.
Yes.
Yeah. So X and Y are not homologous. So there is no recombination between them. And Y is not a possibility. There can be recombination between X and X in females, but not between X and Y. Make sense to you?
Yes, sir.
Yeah. Okay. 57. Will you read that, Atif? Yep.
Yes, sir. Plasmid has been used as a vector because option A, it is circular DNA which have capacity to join to eukaryotic DNA. Option B, it can move between prokaryotic and eukaryotic cells. Option C, both and show replication. Option D, it has antibiotic resistance gene.
Yes, Atif, that's correct. Correct, Hiba. Right. It's A. It is a circular DNA which have capacity to join to eukaryotic DNA. That's where we can insert our gene of interest, which is a eukaryotic gene or a eukaryotic DNA. And that's why plasmids can be used as a vector. Perfect. So a couple of more questions we'll take, which are more conceptual. Is 64 is again a repetitive question. It's easy. It talks about restriction enzymes. And there's one more question. Mm, yes, 58. Will you read that, Atif? 58? This is the last we'll take.
Yes, sir. The process of replication in plasmid DNA other than initiation is controlled by option A, mitochondrial gene, option B, plasmid gene, option C, bacterial gene, option D, none of these.
Mm-hmm. What do you think? Okay, Heba is correct. So the right option is bacterial gene. The question again, it's a simple question. So if you look at it, the process of replication in plasmid DNA, other than initiation. Initiation is important and it is only done by ORI, right? Origin of replication, right? That we have to put, correct? That is plasmid's
Yes.
own gene. Rest of the process of replication, which requires proteins, for example, DNA polymerase, that all is given by bacterial gene. Okay, plasmid does not have a gene for DNA polymerase or other proteins that bind to the plasmid and do the replication. So only initiation, ORI. Remember the four things were there. ORI was there, and what else? Selectable marker was there. Our gene of interest was there, and cloning site, the site at which restriction enzyme will cut was there. So most of the rest is given by the bacterial gene because plasmid is present in the bacteria. Make sense? Atif, Anima?
Yes.
Yeah. Okay, cool. So we'll stop here for today. And tomorrow I'll not tomorrow in the next class. So we have a couple of more classes which are stipulated for revisions. So we'll go to applications of biotechnology first, and then to some other important concepts. Okay, of reproduction. Yeah. Okay.